Hi, everyone. I'm happy to participate in this lab automation presentation focusing on microfluidics. I'm Shiva here. I'm a professor in the Department of Chemical Engineering. I'm also a CEO and co-founder of NemaLife, where we are trying to commercialize this high throughput screening platform using the animal model C. elegans. So in terms of the outline of my presentation, I'm just going to give you a quick overview of C. elegans and some of the challenges in terms of drug discovery. And then I'm going to go into technologies that have been used for high throughput C. elegans screening, and then focus on some specific examples that will give you impression on how data in C. elegans can be used to translate to human studies. And then I'm going to talk about two specific projects. One is focusing on doing a drug discovery screen using an Alzheimer's model of C. elegans. And the second one is to very exciting project looking at how C. elegans could be used to do studies, in vivo studies on the International Space Station. Again, this involves fluidics as well. And then I'm going to end with some summary and outlook. So with that, I'll jump into the challenge. So as you may know, this is the typical drug discovery pipeline where compounds are being screened for efficacy and safety using in vitro assays as well as assays in rodents. And the choice of these compounds has been extending significantly in recent years simply because of advances in microbiome and AI, in which cases the size of the library could be actually be large. So at some point, some downstream assays need to be done in terms of screening to evaluate the safety and efficacy and identify the lead hits. So this process of preclinical R&D can take up to 3 years. And as you may know, with in vitro assays, they do have some limitations, although these are being addressed as we speak using organoids and organ on a chips. But still, these tools are not sufficiently advanced, according to me, to have the complexity which is important in terms of drug discovery. But alternatively, if you want to do testing in rodents, the problem is that it's slow and expensive, and it's not really scalable because it's hard to do or very expensive to do screening with hundreds of compounds. And then and then on top of it, there's also ethical concerns of doing animal testing. So these are some of the challenges which the drug industry is seeing, especially in the preclinical phase. And sometimes there's always an iterative loop of doing in vitro testing and mice as well. So our proposition is to really start thinking about, could we get in vivo data very early on in the drug discovery process? And that's where the use of C. elegans could be an important tool to de-risk screening, right? And because it's an organism, it's an intact whole organism, then it clearly addresses some of the limitations of in vitro assays. And in fact, I would say that the throughput of C. elegans testing is becoming comparable to in vitro assays. That means it is possible to start thinking about in vivo assays early on, because as I'll show, it's a very scalable approach to do high throughput screening. In fact, in the startup over the last 2+ years, we have already replaced the use of 500,000 of mice because our partners have chosen to do high throughput screening in C. elegans, and that has led to enormous savings in R&D costs. And our goal is to keep pushing this, where the idea is to get in vivo data early on and just have rodents or mice testing as an option or an essential tool because of safety requirements, and to obtain the PK and PD information that can be used to start thinking about the clinical trials in humans. So this is a powerful approach. In fact, C. elegans has been used in academia for almost half a century. And this broad information base, along with the novel tools, eh, uh, really allows us to start rethinking about how to do high throughput screening in C. elegans. So a quick primer on C. elegans itself. It starts as an egg, and in a few days it becomes an adult. And after it becomes an adult, it enters into the reproductive phase, and the reproductive phase is typically about 6 days. And then a single adult can lay up to 300 eggs during this period of 6 days. So that means the brood size is large, so it gives you, it makes essentially breeding easy, because now you can get a high number of animals to do your screening. So that's a significant advantage of this portal organism. And as you see in this video here, the great thing about the worm is that it's transparent. It lives only about 3 weeks, and the basic pathways are conserved between C. elegans or worms and mammals. So that means the essential biology is preserved. And on top of it, as I was mentioning earlier, the community of C. elegans has developed strains and reporters, and also disease portals. So this becomes a powerful like tools that you can use in terms of drug discovery as well. And especially in terms of space flight, C. elegans offers enormous advantages because of the small upload mass and the small footprint which is needed to do experiments on the space station. So all these factors are culminating to rethink how to use C. elegans in the drug discovery pipeline. And in fact, the FDA has also been thinking about this, and there's already significant papers on this topic, especially in the context of toxicology. So I would encourage you to look at these papers, because it's showing increasing evidence of ability to translate data from C. elegans into animals. And there have been significant breakthroughs, a few of which I've highlighted here, including missions to the International Space Station. And because it lives only 3 weeks, there's a significant number of these compounds have been discovered that extend the animal life. So the whole aging field has been propelled because of this organism, and therefore there's significant number of papers that are being increasingly being published in this field. Um, so now this entire body of knowledge is now a prime time to start thinking about screening technologies. Um, Now, this is how screening has been done in C. elegans in the past, where the standard medium is to use agar plates, which you see on the left here. The challenge with using agar plates is that because of that reproductive phase, which I discussed earlier, the animals have to be transferred daily, and that imposes challenges in terms of labor and throughput. And another big drawback, especially for drug screening using plate-based assays, is that a significant portion of the drug is just soaked in the agar. So therefore, the animals are not getting enough dosage, if you will. So therefore, it actually complicates interpretation of the data. However, the good thing is that the animals are living on the surface of agar. They're experiencing the ambient conditions. So therefore, oxygen and transport of gases is not a limitation when doing plate-based assays. On the right is probably the technology which is more typical of drug discovery industry, if you will, where worms are loaded in these wells. But again, here, because of the reproductive phase, unless you use some special drugs to block reproduction, it really becomes limiting to do whole life. That means studying older animals and so on. And the reason it's important to look at older animals, because most diseases are linked or associated with age as a risk factor. So that means all the phenotypes which you see because of aging are difficult to do in these standard plate-based assays. The other issue with these plate-based assays, with these hydroplates is that if you don't do aeration, then it can create a hypoxic environment. So these are some of the limitations which have really stifled the field to do high throughput screening in a clean way to especially get whole life data. And in terms of our work, we have used the previous existing body of knowledge in terms of the strains and the disease portals and the reporters. But now we are introducing two new technologies, if you like. One is using microfluidics as a way to control the animal environment and also do assays in parallel. The approach which we use involves taking lots of videos of animals in these fluidic chambers. So therefore, you need a way to analyze images, detect all the phenotypes. This is where we are using AI as a tool to essentially do automated image processing and extracting data out of these videos. So this is the entire pipeline, which involves using the this organism and the microfluidic automation and AI to do the analysis. So here is a quick video that shows how the actual system works. So I'll play this and I'll stop at different places to explain kind of the key pieces of the technology. So the this initial segment is showing that it's very tedious to pick animals on agar plates. But now this chip design, which you see here, it's a very simple design which has a single inlet and a single outlet, and animals are loaded at the top of the chamber. And the rectangular chamber that you see, we introduced two very novel features that allows us to eliminate all the tediousness that you see in terms of the plate-based assays for moving animals around. And the two features are one, so this is a high resolution SEM image of the chamber. And you can see that the first feature are this array of these pillars, which essentially allow the animals to crawl, unlike animals thrashing in liquid, and that creates a stress-free environment. The other key engineered feature are these blocks that you see, which where the spacing is optimized to allow the eggs and progeny to be removed. However, the adults are still trapped in the chamber. So that means that these blocks allow us to do all the filtration, if you will, of progeny during that reproductive phase I was discussing earlier. So, and we can make these, hundreds of these chips, allowing us to do high throughput screening. And here is a segment that shows how we're able to remove the progeny and essentially automate animal husbandry at scale. So the worms are all shown in this image here, but you see these gray specks, these are the larvae or the progeny. And by just doing a simple wash from the left, you will see that the fluid is able to remove all the progeny, but the adults are practically staying in the field of view. This is really the core piece of the technology that automates the animal husbandry. And these videos are now uploaded on the cloud, and the AI is auto segmenting worms and scoring all the phenotypes. So this is essentially the pipeline which we use for high throughput in vivo screening. So we operate this in two ways. One is, as I described, where we take these bright field images and allowing us to get data from adults all the way close to their death. So this whole life screening platform is a powerful way to look at age-related disorders. At the same time, because the chips are transparent, we can take those chips, and if the strains have fluorescent reporters, either GFP tags or mCherry tags, we can use a high content imager, do epifluorescence imaging, and look at actual tissues and cells to understand the mechanism of action. So that means by integrating both the organism level readouts as well as looking at the fluorescent features at the level of organelles or cells, we can get a significant amount of depth in the phenotypic space to understand how these drugs are actually acting in terms of the pathology which you are interested in. So because we are able to grow these animals across life, the initial checks we did were with this anti-aging compounds. As I mentioned before, there's a significant number of these compounds which have already been published. So we took half a dozen of them and tested them in the fluidic platform. And you can see, as expected, all of these compounds are able to show significant extension in animal life, indicating that the chip environment and the way the fluid processing is done is still able to show the efficacy effect which is observed on plates. An interesting thing that we see is that the dosage required to see this efficacy is lower in the chips as compared to on plates, because as I uh, remarked earlier, a significant portion of the drug is soaked in the agar medium. Therefore, the dosages are much higher on plates, whereas in the chips, there is efficient uh, delivery of drugs, so the dosage is smaller. The other example which I would like to highlight is not from our work, but is from the industry. Say ADM has done a screen looking at essentially anti-obesity. So the idea is to look at probiotics which can reduce the accumulation of, 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 of lipids or adiposity, basically. So they did a screen in C. elegans and identified the hits, and they showed that these hit could also show similar efficacy in rodents and humans. So this is a very nice example where discovery in C. elegans has led to an actual product on the shelf. And the other example which I'm showing here is also from industry, which is now focused more on supplement that was uh, which helps to improve the function of muscles in the elderly. Again, the initial studies were done in C. elegans and rodents, and the mechanism of action was all identified, and the clinical studies show that it was translatable to actual people. So this is again a product on the shelf. So these two industry examples highlight that discoveries in C. elegans could be translated to studies in humans. Now, the last example I want to give in terms of the translatability is a study which we did in a disease model of dystrophy. Um, and the idea here was that the FDA-approved drug prednisone is an effective treatment for this disease. So then the open question was that would prednisone also rescue muscle function in the disease in the disease model in C. elegans. So we looked at the strength and the swimming prowess, which you see here, and we indeed find that treating worms with this drug indeed improves the function of the muscles, indicating that drug that is being used in humans is indeed translatable to C. elegans. Uh, so now I'm going to talk about two specific projects. One is how we used the Alzheimer disease model to do a rapid screen in C. elegans. And the second study that I'm going to talk about is the spaceflight project. So the basic hypothesis that was underlying this screen was that, as I indicated earlier, uh, in terms of AD, age is a risk factor. In fact, the prevalence of AD doubles every 5 years if you're older than 65. So that is a significant risk in terms of seeing AD in older people. So the hypothesis we had was that given that the literature already has these anti-aging compounds, could we repurpose those for AD? So then we tested these anti-aging compounds on C. elegans, which you see on the pie chart here. A significant fraction of them have been discovered in C. elegans. So then it was easy to take those compounds which extend animal life and tested on Alzheimer models of C. elegans. So the way we did that was, first we did the screen in cow animal model, and we found that out of the 250 or so anti-aging compounds, about 100 had improved uh, animal survival. And then we did another screen to see how much we can replicate those, and about 35 of them have been replicated in this tau model. And finally, we took those hits and retested them in an independent Alzheimer or A beta animal model, and we saw that a significant number of hits still persisted. So that means that this entire pipeline where we used two animal models and tested 250 drugs was accomplished in just 3 months. This would practically be impossible to do these kinds of AD studies in mice. So hopefully this shows the speed and the power at which you can do screening in C. elegans. And then subsequently, we've been doing uh, 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 RNA-seq studies to understand the genes which are differentially expressed. So this gives insights into the mechanism of action. And I would like you to draw your attention to the figure which is in the middle, where NL1, NL2, NL6, NL9 are the hits in terms of our screen. And we also show the number of genes which are differentially expressed in terms of the overlap with these different drugs. And if you look at NL1 and NL6, there's hardly any overlap in these genes which are differentially expressed, indicating that NL1 and NL6 could be acting on different pathways. So this really allows you to do rationally combine drugs and develop pills for AD. So this is a very unique way of thinking about drug screening and combination screening. And the last few minutes, I just wanted to talk about our second project, which is to how do we do space biology studies on the International Space Station. And here, as you may know, astronauts lose a significant amount of muscle mass uh, on the space station. And if the goal of NASA is to go up to Mars, which takes up to a year, this could become a significant obstacle. So there's a need to deeply understand how the muscle physiology changes, and then hopefully develop treatments which could address that. And C. elegans is a low cost experimental animal model which allows you to look at these things. However, the challenge was that we even though previous studies have shown that the genes which are present in the muscle are differentially expressed compared to Earth and space flight, but the hypothesis of whether there's actually a decline in the strength, it was not known. So we developed a microfluidics based approach to look at this. And as I mentioned, just like in terms of biology, there's options to choose different animal models, and all of them have been used in different studies in space biology. In fact, C. elegans is probably the organism which has been flown enough number of times to the space station, as you see from this table. And as I was saying that our goal was to look at the muscle strength in C. elegans. To develop that, we have engineered a device. Again, we use the idea of pillars, where now in this case, the pillars are dangling inside the chamber. Because they're dangling, the worm as it crawls, it's able to deflect them. And by recording how much deflection happens in these pillars, we're able to extract the forces which these animals are able to exert. On the left is showing how we load animals into the device through this narrow neck, allowing us to have only an individual or two inside this chamber, which allows us to record its motion as it's deflecting these pillars. On the top right is the flight hardware, which was flown to the International Space Station, and where the astronauts they were able to inject the worms which were in the bag into the fluidic device, and then take the device and do imaging on an inverted scope. And our initial results show, which is shown on the plot in the lower right, you can see that at time point 2, animals are indeed becoming weaker. So this indicates that the C. elegans and the NemaFlex assay, which is the device that we use to record the strength of the worms, is able to capture the basic physiology which is observed in astronaut, which is loss in strength. Um, so hopefully this gives you an impression as to how fluidic devices could be used to do in vivo testing also on the space station instead of only using rodents. So with that, I would like to stop here, and I would like to thank all my collaborators who've been helping us with the C. elegans projects and assays, including providing reporters. And also would like to acknowledge the team from NemaLife, who have been instrumental in really translating this technology out of an academic lab onto an industrial scale. And then, of course, my students and postdocs who have been doing C. elegans studies and optimizing devices and doing screening. So I appreciate all their efforts in terms of developing this high throughput in vivo screening platform. So I'm going to stop there. And again, if you have questions, please email me. I'm very active on LinkedIn and so on. So I'll be happy to take any questions offline. So thank you.